Okay, guys, we'll go ahead and get started here. So today we're going to talk about enzymes as drug targets. So this is kind of a standalone lecture, and I'm going to go ahead and turn off the front lights here so you guys can see this a little better. So this is sort of, like I said, a standalone topic, but there are a number of drugs that actually target enzymes instead of receptors. So what we're going to do today is give you a little bit of background, kind of a refresher on how enzymes work. And you need to have that information in hand in order to understand the pharmacology of drugs that target enzymes. Because enzymes are not receptors, they're different. Enzymes, of course, are catalysts. They actually do chemistry. They catalyze chemical reactions. But they can be great drug targets. So the first the first 15 minutes or so, we're going to talk about enzymes, just refresh you a little bit on enzymes, what they are and how they work. And then we're going to get into the drugs that target them, which for the most part are called substrate analogs, by the way. Okay, so the objectives today are to review the basics of enzymes, and then we're going to talk about a number of drugs that actually target enzymes, bisphosphonates, non-steroidal anti-inflammatories, kinase inhibitors. I'm also going to mention statins. And then we're going to, another goal is you should be thinking about the molecular target, the cellular response, and the effect, like we always do. And we'll get a little bit, there'll be a little bit about biochemistry, but not too much. So what are enzymes? Well, they are catalysts. They're biological catalysts. And so they're basically a catalyst is any substance that increases the rate of a chemical reaction and itself is not changed by the chemical reaction. So an enzyme fits that definition, but the terminology you really need is really right here on this slide. Definitely I want you to understand what a substrate is and what a product is. So the substrate is some chemical compound that's going to be acted upon by an enzyme, and when that enzyme acts on that compound, it alters it chemically. In this case, it cleaves a single molecule into two different molecules. So the substrate is the chemical compound before catalysis, and the products are what you get after catalysis. You could also have the substrate be two different chemical entities, and then the enzyme links them together. But anyway, the substrate is before and the product is after. So what happens, an enzyme typically fits together with the substrate kind of like a lock and a key, just like a receptor and a ligand, for instance. But there's a difference. Once the substrate binds to the enzyme at a place typically called the active site, it fits together kind of like a lock and a key. And you get this enzyme substrate complex. And sometimes the enzyme actually changes shape very subtly when the substrate binds to become active. And then the enzyme works, it facilitates the catalysis, and the products leave. Now, this reaction of going from the substrate to the products can occur without the enzyme, but it might happen so slowly that it almost never occurs. So the enzyme oftentimes facilitates a chemical reaction like this. So there are different modes at which different enzymes bind their substrates. The one I showed you is really kind of like lock and key. The enzyme and the substrate just fit together perfectly. But sometimes there is a kind of a process called conformation selection. So the enzyme is typically a protein, and if we're thinking in terms of like molecular variants, even quantum mechanics, all sorts of things, the enzyme can be constantly fluctuating between subtly different conformations, different structures. And in some cases, people have found that when the substrate binds to the enzyme, it locks it into an active state. Okay, so the act of substrate binding changes the enzyme, stabilizes a particular structure of the enzyme, which is then active. And that mechanism is called conformation selection. And there are other cases where what's going on is called induced fit. The enzyme never quite has the active structure, but once it binds the substrate, that interaction induces the enzyme to change its structure slightly, and now it can become a catalyst. So those are the three general modes by which different enzymes work: lock and key, conformation selection, where you're selecting a particular conformation out of many possible conformations, and induced fit, where binding of the substrate to the enzyme actually changes the structure of the enzyme a bit. And each particular enzyme will typically work by one of these three mechanisms, depending on which enzyme it is. And you can see here in this pie chart that most enzymes are lock and key type. A few of them are conformational selection, an appreciable number, maybe 15%. A very few are induced fit. And then there are some enzymes that have a little bit of a combination of two or more of these kind of mechanisms. So enzymes do many different chemical reactions, and I'm just going to put up two slides that will show you a handful of the most prevalent classes of enzymes, families of enzymes, if you will. So there are oxidoreductases, and these are enzymes that catalyze oxidation reduction reactions, which means they're transferring hydrogen and oxygen atoms from one substance to another. So here's an example here where a hydrogen atom is transferred from A to B. Here is oxidation. Then there are transferases, where they transfer a functional group from one substance to another. So not a hydrogen and oxygen, but a small organic group like a methyl group, an amino group, the phosphate group, etc. So it just basically transfers something from one compound to another. So compound A has B attached to it, and the enzyme connects that to C. Hydrolases form two products. So you have a compound that has an A half and a B half, and then these get split apart. That's kind of like the cartoon I showed you initially. So those are hydrolases, and they're oftentimes called hydrolases because often water is liberated during that chemical reaction, okay? So oftentimes when you're breaking apart molecules, H2O is eliminated. But hydrolases split one thing into two things. Lyases basically add or remove groups or substrates, but it's not water is not eliminated by those. Isomerases actually change a chemical structure from one isomer to another isomer. So the chemical formula is the same in the product as the substrate, but it's structurally rearranged. And then ligases put things together. They join two molecules together, okay? So they connect things together. You see this in polymerases, for instance. So anyway, these are just the general classes of enzymes. There are more, but these are some of the more common ones. So just become familiar with those. Okay. So I also want to mention that enzymes oftentimes have cofactors associated with them. And usually this is a metal ion of some kind or another. So a lot of enzymes at their active site have like zinc or iron or copper, magnesium atom that's held in the active site of the enzyme. And the reason this is so common is that these metal ions can undergo different oxidation states, so they're involved in electron transfers and things like this. And here's some examples of some of these, okay? So for instance, DNA polymerase has a magnesium ion in it. Cytochrome P450s have iron. So does hemoglobin for that matter. zinc atoms are found in carbonic anhydrase, alcohol dehydrogenase. These are just various and sundry examples. So cofactors are critical parts of the enzyme, but they're not the enzyme itself, because enzymes are proteins, right? So they have genes, they're encoded in DNA, then you make RNA, then you make the protein. But of course, the genetic code doesn't specify for metals to be included in the protein. So we consider these cofactors. And here is an example, DNA polymerase. And so this is part of the structure of DNA polymerase. And you can see that it has actually 2 magnesium ions present in the structure. And that's going to typically be right near the active site of the enzyme. Okay, so cofactors are oftentimes these metal ions, but some enzymes also use something called coenzymes. So coenzymes are molecules that participate in the chemical reaction itself. And cofactors cannot be used up. They just change oxidation state typically. But coenzymes do get used up in the reaction. And so you've probably heard of some of these important ones like NAD, NADH. And here is NAD and here is NADH. And what it is doing is it is a reducing agent. It's a source of electrons, okay? So typically, these coenzymes get together with the enzyme and the substrate, and they do something chemically, but they're chemically altered when they do their job. And so oftentimes there are pathways in the cell that will regenerate the active coenzyme by a separate enzyme. Here is coenzyme A and acetyl-CoA. And once again, this is a coenzyme. And in this case, acetyl-CoA, for instance, it's a source of carbon atoms. So acetyl-CoA is involved in a lot of biosynthetic reactions where you're building up complex carbon-containing compounds, and it's actually contributing carbon atoms to a growing chemical that's being synthesized through biochemistry. So obviously, if you contribute carbon atoms, you're different after you contribute carbon atoms, right? So this has to be regenerated in order to become active again. So coenzymes are important. They contribute to the chemistry of the catalytic reaction that is mediated by an enzyme. I'll talk just a little bit about energetics of enzymes. So if you think about it, here's kind of the free energy level of the reactants. This is also, we would call this the substrate. And here is the free energy of the products. So you can see that the energy of the products is lower than the energy of the substrate, the reactants here. That means this is an energetically favorable chemical reaction. It can go all by itself, but it might, and the degree of favorability is measured by delta G, the difference between this level of energy and this level of energy. But oftentimes there is basically an intermediate state that has to form in order for the reaction to go. And that is usually a higher energy state. So even though this is a favorable reaction in terms of comparing the free energy of the product, of the reactants and the products, it may go very slowly because this energetic hill you have to get over to make it can be substantial. And so enzymes reduce that energy, and now it might become favorable, and this reaction might go 1,000 times faster with the enzyme being involved. So that's basically the definition of catalysis. The enzyme is acting like a catalyst, and it's lowering the activation energy so that the reaction can go forward at an appreciable, biologically relevant rate. Okay. And of course, you know, there's a forward rate and a reverse rate. Okay. So some enzymes stabilize an intermediate state of the reactants, and so in this, and some don't. But in this case, there's an intermediate state where the chemical reaction that's being catalyzed is partway completed, and you can actually get a great increase in the rate of the reaction by certain enzymes that stabilize the intermediate rate. But mostly I want you to just kind of know that, you know, this is energetically what's happening to enzymes. For the most part, they are just reducing the activation energy needed so that the rate of the reaction can go forward much more quickly. So there is kind of a famous area of biochemistry called Michaelis-Menten kinetics, which I'm not going to go into any great detail here. But if you think about it, this is kind of the steps of what has to happen for a chemical reaction to occur. You have an enzyme and a substrate. Remember, that's the reactants. The first thing that has to happen is the enzyme and the substrate have to associate with one another. So the first step is association. And then once the enzyme and the substrate are together, then catalysis has to happen. And you go from a substrate to a product. Now, it could be taking something apart, putting something together, but those are the steps. So it's a little bit different than, for instance, a ligand or a drug binding to a receptor. There, the only thing that's happening is the first part, association, right? In the case of enzymes, association is only the beginning, and then once the enzyme-substrate complex is formed, then some sort of a chemical reaction has to occur. So there are rate constants involved. There is K1, which is the association of enzyme and substrate to make this enzyme-substrate complex. But then this can also dissociate, and that's K minus 1, okay? Once the enzyme-substrate complex is formed, then there is another rate at which that substrate is then catalyzed and changed into the products, okay? So there's kind of a two-step thing going on, whereas a drug binding a receptor is only a one-step thing, okay? So that's a little bit different. All right, so that is like the whirlwind tour of enzymes, the basics, but that information sets us up to understand some pharmacology, okay? Questions on that so far? It's a good time to ask. Pretty clear? Okay. Okay, so let's get into some drugs that target metabolic pathways, and you'll get to see how these work. And many drugs are actually what are called substrate analogs. So we know what a substrate is, right? We just talked about it. That's the thing that ordinarily is acted upon by an enzyme. A substrate analog is a chemical that looks like a substrate, but it's not. It's an analog, which means something that's similar but different. And so basically, substrate analogs are chemical compounds that have a chemical structure that resembles the substrate. It almost looks like a mimic. And it can associate with the chemical, with the enzyme at the active site typically. It binds to the active site, but it's an analog, and typically it cannot be metabolized. It cannot undergo a reaction. So what's that going to do? The similarity is going to allow the substrate analog to bind to the enzyme, but then because it's something different than the substrate, it plugs up the enzyme because it can't be catalyzed. Okay, so it's going to typically be an inhibitor of the endogenous substrate. And the substrate analogs are the most common metabolic inhibitors in terms of drugs. So I think this becomes clearer if we go through a specific example. And the first one I want to mention is a very important drug, this class of drugs called statins. So statins decrease cholesterol, and they're among the most commonly prescribed drugs out there. because a lot of people have high cholesterol and they have risk of cardiovascular events, and you need statins to prevent their cholesterol from getting too high. So before we talk about statins, I want to just walk you quickly through this biosynthetic pathway. This is an anabolic pathway. It's a pathway that builds up cholesterol. And you can see that it starts with acetyl-CoA. That's a cofactor, right? What happens is acetyl-CoA is supplying carbon atoms. And then you make HMG-CoA, and then mevalonic acid, mevalonic diphosphate, farnesyl pyrophosphate, something called squalene, lanosterol, and a few more steps, and you end up as cholesterol. And what you can see is this biosynthetic pathway is building up cholesterol. So your body makes cholesterol, and it's made in liver cells, in hepatocytes, and the amount of cholesterol in your blood is a mixture of cholesterol that you yourself have made in your liver and cholesterol that you get in your diet. Okay? And more is actually made than come in by your own body than comes in through your diet. So if somebody has high cholesterol, one pharmacological approach is to inhibit their ability to make their own cholesterol, right? And how do you do that? Well, it turns out that, let's look at the first two. Each of these chemical changes is mediated by enzymes, and only two enzymes are shown here. HMG-CoA synthase makes HMG-CoA, and then HMG-CoA reductase turns HMG-CoA into mevalonic acid, mevalonate, and that will eventually form cholesterol. It turns out that each of these biosynthetic steps has its own rate, and the slowest one is HMG-CoA reductase. So that's kind of like a bottleneck anyway, because that's HMG-CoA reductase is sort of a slow enzyme. And so it turns out if you inhibit HMG-CoA reductase up here in this pathway, everything downstream is going to be formed at lower rates. So you inhibit this enzyme and you inhibit the formation of cholesterol. And that's what statins do. So statins must be some sort of an inhibitor of the enzyme HMG-CoA reductase. Let's have a look at the structure of statins and compare it to like HMG-CoA. So here is the substrate for the HMG-CoA reductase reaction. Here's the product. Here's the structure of a statin. So here's basically, you'll notice that there's a big chemical resemblance, right? Here is the structure of a statin, and there are many of them, and they have different R groups, which can be any one of a number of different things. And here is HMG-CoA. Very, very similar, right? So it's a substrate analog because it's similar to the substrate, okay? Chemically, but it cannot be acted upon by the enzyme. So it's going to plug up the enzyme at the active site. And so this region that is similar, that makes it a substrate analog, this portion of the statin is called a pharmacophore, and that's a very important term. It is a chemical structure that resembles something else. It's also the chemical structure that is the business end of the drug. It's the important structure. So the pharmacophore of statins looks a lot like HMG-CoA. And this is indeed a substrate analog. And you can see that chemically here. So here are a couple of statins. Here's this R group that's present in statins. So here's lovastatin, for instance. Right there, that is the pharmacophore there. And here is the pharmacophore in rosuvastatin. They're different drugs, but they have similar pharmacophores. And there's a resemblance between the pharmacophore and the normal compound. And that makes some substrate analogs. So what's really happening is in this pathway, if HMG-CoA reductase binds a statin instead of HMG-CoA, that enzyme is now out of action. And you're not going to make the, you're not going to continue with that pathway leading to cholesterol. Make sense? Kind of cool, huh? You trick the enzyme into grabbing something that looks like the right thing, but it's not. And that's how these substrate analogs work. Here's another example. We'll go through some examples. More substrate analog stuff. So bone is very interesting. Bone is constantly being made and constantly being resorbed. And it's the balance of making new bone and resorbing bone that determines how much bone there is. In elderly people, they can experience osteoporosis. And that means that basically that balance of making new bone, replacing bone, and destroying bone is out of whack, and you lose bone. So bone contains calcium phosphate. It's basically made out of calcium phosphate. And bone density itself comes from the balance of formation and resorption. And this balance can kind of get out of whack with age. So here's what's ordinarily going on, especially at the end of long bones, where arthritis can occur. Basically, there are two types of cells that do this. One is called an osteoblast, and the osteoblast makes new bone. And then there are cells called osteoclasts, which actually resorbs bone. It removes bone. What bisphosphonates do, these are drugs, they inhibit bone resorption. So if you inhibit bone resorption, more bone is retained, right? And so that's how these drugs called bisphosphonates work. And they can treat osteoporosis, which is a big problem in geriatric medicine. Leads to a lot of broken hips. So one of the chemical pathways that participates in the bone resorption process is the following. So it's a complex procedure, but there's an enzyme called farnesyl diphosphate synthase. And basically, it looks like this. You don't have to remember this in detail, but what I want you to see is that there's a lot of diphosphate involved in this, right? Diphosphate here, and this is free diphosphate. So farnesyl diphosphate synthase, this enzyme participates in bone resorption, and a class of drugs have been developed called bisphosphonates that are also substrate analogs. So here is diphosphate. Diphosphate also goes by the name pyrophosphate, same thing. A bisphosphonate looks similar to that, right? You got two phosphates, but instead of having an oxygen in the middle, you got a carbon that can contain a couple of R groups. So a structural similarity, right? So the bisphosphonate has a similar size and structure to the diphosphate. It's a fake, it's a mimic, and it's a substrate analog. And so here are a number of different drugs. They're all bisphosphonates, and you can see that they have a carbon atom between the phosphates instead of an oxygen, and that things are linked up to them. But these are all substrate analogs because they look very similar to pyrophosphate or diphosphate, whatever you want to call it. So again, it's a resemblance. Okay. So these things actually can increase bone mineral density. And here you can see like some clinical data. So here we're treating people over a period of months out to 3 years. And here's the placebo down at the bottom. No real change over 3 years. And here on the x-axis is the percent change in the bone density in the spine of the lower back. And what you can see, whoops, as I drop my microphone stuff here, what you can see is that these drugs have an effect because it's different than the placebo. There we go. So once again, here's the placebo. And here you can see some of these bisphosphonates up here that increase bone density. Okay. Some of these are not bisphosphonates, like raloxifene, for instance. Turns out that a lot of bone density is also influenced by estrogen. So one of the reasons that osteoporosis is a problem in postmenopausal women is that estrogen levels go down, and estrogen also feeds into this pathway. So you can actually get some bone density improvement by hormone replacement therapy as well. But we're talking here about bisphosphonates. So anyway, you get the idea. But that's the sort of experiment of how things are done in clinical trials. You have to compare how well somebody does compared to a placebo over time to see if it works. Another example would be COX inhibitors. So COX inhibitors actually inhibit cyclooxygenase, and there are a bunch of these. So what's up with cyclooxygenases, abbreviated COX? So there are two of them, actually, two enzymes called COX-1 and COX-2, and they have very similar activity. They're both cyclooxygenases, but they have different expression patterns. So they're expressed in different places in the body. And here's the deal. COX-1 is constitutively expressed in most tissues. That means it's kind of a generalist. This enzyme is present in almost all of our tissues. But COX-2 is inducible, and it can turn on. But the key here is it's expressed primarily in macrophages during inflammation. So macrophage are immune cells that can phagocytize things, but they participate in the process of inflammation. Okay. So COX-1 is present in most tissues. COX-2 is kind of a specialist enzyme. It's expressed primarily in macrophage. But if you inhibit either COX-1 or COX-2, you're going to reduce inflammation by a mechanism that involves prostaglandins. So here is a diagram here showing you a little bit of how this works and how it ties in with inflammation. It turns out that a compound called arachidonic acid, it is metabolized and produces compounds called prostaglandins. And that changing of arachidonic acid into prostaglandins, one of the steps involves either COX-1 or COX-2, okay? Now it turns out that arachidonic acid also does other things. It can be used to, some of the products actually lead to the production of pro-inflammatory cytokines and things like that. But the COX-1, COX-2 stuff, we're really concerned with turning arachidonic acid into prostaglandins, which cause things like inflammation and pain and swelling. So by the way, inflammation, pain, and swelling, they're unpleasant to experience, right? Everybody knows that, but it also tells you something is wrong. So if you didn't have inflammation, pain, and swelling, you could have joint injury and not even know it, right? So it serves a purpose. And inflammation itself also has some, even though it's unpleasant, has some repair type activities because it recruits immune cells that might remove materials or pathogens that are present. So, pain and swelling are unpleasant, but there's a reason it's evolved. But sometimes inflammation can go way out of whack and there's a hyper response. And that's when these COX inhibitors come in, when you want to kind of tone down a normal response that's kind of gone out of control, resulting in pain and swelling. So now we'll get into the pharmacology. And I'm never going to ask you to remember and study how a biosynthetic pathway like this works. But what I will perhaps ask you to do is to understand what's going on with it. I don't have this committed to memory. I don't like wake up at 3 a.m. and go, oh, the arachidonic acid metabolism pathway. You know, but I know how to interpret these things, and so I just want to help you do that as well. So let's look at this. Arachidonic acid is up here. Prostaglandins are down here. And this is showing all of the enzymatic transformations in a pathway leading from arachidonic acid to prostaglandins down here. And you can see that it's just a multi-step thing, you know, similar to the cholesterol pathway I showed you, but different compounds involved, step by step. Parts of it are linear, parts of it are branching. But the enzymes are shown in red here. And so here's COX-1 and COX-2 right here. It's turning this beast into this beast, which then can lead to prostaglandins. So what happens if you inhibit those enzymes, COX-1 or COX-2? Less prostaglandins, right? Okay. And so, not that tough to interpret, really. So aspirin and ibuprofen are COX inhibitors. And sometimes prostaglandins can be used as drugs too. But at any rate, these are cyclooxygenase enzymes, COX-1 and COX-2, and they catalyze the oxidation of arachidonic acid. They also happen to be oxidoreductases. But here is the real reaction here. Arachidonic acid eventually using COX-1 and COX-2 leading to PGH2, I think that's prostaglandin H2, kind of simplified. But you remember I told you that COX-1 and COX-2 are expressed in different places. You know, COX-1 being broadly distributed in the body, COX-2 specialized in macrophage. So, and COX-1 and COX-2 are two different enzymes, even though they do a similar thing. So different drugs can be developed that can either target COX-1 but not COX-2, and other drugs will target COX-2 but not COX-1, and then some of them don't really care, they inhibit either of these. And so here are these different structures of COX inhibitors, and it shows you whether they're more selective for COX-1 or COX-2. So remember selectivity from last time? In this case, we have one drug with two different possible targets, and we want to see which drug is more selective for COX-1 versus COX-2. And let's take like a couple of favorites. Aspirin. Where is aspirin on this list? It's up here. So on the left, the more blue is more COX-1 selective. If it trends toward yellow, it's more COX-2 selective. And aspirin has some preference for COX-1 over COX-2, so it's moderately selective. Here are some down here that are especially selective for COX-2, okay? How would you measure all of these different selectivities? Well, once again, it would be with a KD or a KI, like we've been talking about. It has to do with how tightly it binds to its target, right? So, you know, ketorolac up here has a much higher affinity for COX-1 than it does for COX-2, whereas this one, I don't even know how to say that, lumirocoxib, has more selectivity for COX-2 as opposed to COX-1. And then some are in the middle. And so COX-2 inhibitors tend to be bigger. They have a larger hydrophobic binding product or pocket, but they have different selectivities for these different drugs. Why is that important medically? Well, these ones over here would influence macrophage, and these ones over here would tend to do COX-1, and in the middle is both. So it depends on whether you want to really treat that. You know, these ones over here, since inflammation almost always involves macrophages, these are very potent anti-inflammatories, whereas some of these others up here probably would work on inflammation to some extent, but also just generalized, pain and swelling and that type of stuff. So once again, how do you check out how well a drug works? Well, you do a clinical trial of some kind. And, here is one just looking at ibuprofen, Motrin, okay? So here's the placebo. Basically, they're giving drugs to patients and asking them how much pain they feel over a course of 400 minutes. And you can see that the people who got a placebo didn't have any real change over time. They still experienced pain. But the people who got the ibuprofen, either 400 or 600 milligrams, saw a substantial decrease in pain that persisted for a while, right? Interestingly enough, if you give them placebo, they're at a 70, and then after 45 minutes, it's dropped down to a 55, and that kind of stays. What do you think that is? Why would you get a subtle reduction with just the placebo? Well, you're asking the patients, does it hurt? And they go, no, I took a pill, I feel a little better. So that's the placebo effect, right? Which is appreciable, actually. Okay. All right, so last few minutes here, 10 minutes or so, since we're talking about enzymes and drugs that target enzymes, we're going to talk about drugs that target kinases. So kinases are enzymes that attach phosphate groups to things. And it turns out that many of the anti-cancer drugs are kinase inhibitors. So we'll get into that a little bit. And once again, I'll give you a little biology mixed with pharmacology here. So typically, cancer is kind of a genetic disorder. And this is like the real quick crash course on cancer to understand this. And so mutations occur in genes, but not just any gene, a gene that controls cell growth, for instance. So for instance, here's some pathways mediated by receptors called IGF, EGF, PDGF, TGF-beta, etc. If you look at this, when these receptors are activated by binding a ligand, an endogenous ligand of some kind, there is all this signal transduction that occurs inside the cell where you're activating kinases and other factors, but leading to the activation of transcription factors that turn on genes involved in such things as cell proliferation, angiogenesis, the formation of blood vessels, and metastasis, the ability of cells to detach and move somewhere else. So those are all cancer things. Fast cell growth, tumors getting vascularized, which is necessary to support their growth, and bits of tumors breaking off and seeding more tumors elsewhere, metastasis. So it turns out that the internal wiring of the cell involves these kinases. And kinases are enzymes that do things like add phosphate groups to other molecules, which oftentimes is a mechanism to activate them. So these cell pathways, you can think of upstream and downstream events. So for instance, this kinase here called RAS is upstream of RAF, and that's upstream of transcription factor activation. This is the most downstream. But these ones here, RAS and RAF, those are kinases, but they're also oncogenes. There are two major types of cancer genes. There are oncogenes and tumor suppressor genes. The one we're talking about right now are going to be oncogenes. So oncogenes, the normal gene is called a proto-oncogene, but if a mutation occurs in that proto-oncogene, it can turn into an oncogene, and it will make an oncoprotein, and that protein oftentimes is a kinase gone hyper crazy. It gets super fast at phosphorylating things and activates a pathway leading to amping up cell growth and stuff like that. So the key members of this are oftentimes oncogenes. There are also tumor suppressor genes that kind of cause cells to kill themselves by apoptosis when they detect rapid growth, but we're talking about oncogenes right now. How do you develop drugs? So these oncogenes are oftentimes serine, threonine, or tyrosine kinases. And let's look at the enzyme. Here is what's going on basically is here is an amino acid in one of these proteins that's involved in signaling, leading to regulating cell growth. It gets turned on oftentimes by adding a phosphate group. And that phosphorylation is mediated by a protein kinase. So, RAF and RAS are kinases that get mutated and form oncoproteins. But the chemical reaction is simply, all they're doing is sticking a phosphate group onto something else, and that activates it. So serine, threonine kinases can phosphorylate serine or threonine residues. Tyrosine kinases phosphorylate tyrosine residues in proteins. And that typically activates them and turns them on. So many of these oncoproteins encoded by mutated oncogenes are kinases, and here are some of the famous ones: Abl kinase, mTOR, RAF, ALK-Y kinase. Abl is like super famous. And these are some of the drugs that have been developed, like imatinib, crizotinib, et cetera, et cetera. And these are drugs that inhibit one of these pathways. So if you look back at this thing here, if you inhibit RAS or RAF, you're not going to have as much production of transcription factors that are increasing cell growth. And that's how these drugs work. But they're basically kinase inhibitors. They tend to come in a couple of different varieties called type 1 and type 2. But here is an example of the active site of a kinase, okay? And the phosphate that a kinase uses, guess where it comes from? It comes from ATP, adenosine triphosphate. It's certainly used in energy all the time, right? In mitochondria. But ATP is also the source of phosphates for kinase reactions. And furthermore, ATP is also a source of energy for many enzymatic reactions. So ATP does all kinds of stuff. It's involved in just general energetics in cells. It's involved in providing energy for enzymes to work. And it's also donating phosphate groups that kinases use to stick onto other groups, and that actually signals that protein to become active. And that's what's going on in cancer. So here's the active site on a kinase, and you can see that it binds ATP. There's an adenine region, a region that binds the sugar, and there's a phosphate binding region, and that's the part that's going to be catalyzed to donate a phosphate. So type 1 drugs kind of bind right where ATP ordinarily would. Type 2 drugs bind in the adenine region, but then they also contain chemical entities that bind somewhere else. And that somewhere else is called an allosteric site. Allosteric means it's not part of the active enzyme core. And there are type 1 and type 2 kinase inhibitors. So imatinib is a good example. It's the first FDA-approved drug that works as a kinase inhibitor, and it's used for leukemia, chronic myeloid leukemia, CML. So in CML, this leukemia, there is a oncogene called BCR-ABL, and it's actually a fusion gene. Part of it is BCR, and part of it is the ABL kinase. And when that translocation occurs on chromosomes, it makes a fusion gene called BCR-ABL, which makes a fusion protein called BCR-ABL kinase. And that's a hyperactive kinase that leads to upregulating growth, cell growth and cell division. So that's why it causes cancer. So imatinib is interesting because it targets a protein, namely BCR-ABL, that's involved in cancer. But it's kind of an unusual case because BCR-ABL is an abnormal protein. It doesn't exist in somebody who doesn't have cancer. So this is a drug that targets a protein that's present only in people with this particular kind of leukemia. So very exquisite understanding of the biology, and the drug targets a mutant protein, in this case a kinase. Here's another one, vemurafenib. And this is a drug that's been developed for melanoma, not as famous as imatinib, but this one actually has a part that binds that allosteric site. But anyway, I just want you to know if you look at the structures, last thing here, if you look at the structures of this drug and this drug, you can see that they have a lot of rings, right? And so they tend to mimic the adenine region to some degree, okay? And that's why they're substrate analogs. So we went through like 4 different examples of substrate analogs, statins, bisphosphonates, these kinase inhibitors, etc. And yeah, so take-home message, substrate analogs plug up enzymes with something that looks like the normal substrate and can't be metabolized, right? Okay, and that's it. And we'll see you again on Wednesday.